has a scientific experience at Jagiellonian University and New York University. Editor of scientific journals, he truly believes that computer science, artificial intelligence especially, will actively help in scientific research. Actually, he already supports drug research as Chief Scientific Officer at Molecule.one. Ladies and gentlemen, let's bid welcome Stanisław Jastrzębski.
Hello. Okay. Hi, everyone. Thank you for the introduction. Thank you for coming. Today, I would like to tell you a few words about how organic chemistry and synthesis planning are important bottlenecks in drug discovery that slow down how quickly we can move a drug from an idea to an actual therapeutic, and also tell you a bit about how artificial intelligence already, at least partially, addresses this bottleneck. Though the main motivation for the talk is to share what I find interesting in drug discovery as a computer scientist. I am not a chemist, and hopefully inspire you to learn more about this field and perhaps even get involved. So first, a few words about drug discovery itself. The first thing you should know is the drug discovery process is both very expensive and very long. So on average, it takes, give or take, 12 years to introduce a drug into the market, starting from an idea, and it costs over $2 billion. It is a multi-stage process that starts with often a stage called virtual screening, in which we are looking through different ideas, and then, well, hopefully culminates in a therapeutic. So here at the end of this process, we have drawn a molecule of aspirin, and we will have to get used to reading molecules. More on that in a second. Interestingly enough, drug discovery process is not only expensive, but it is also increasingly expensive. So this plot shows on x-axis year, and on y-axis the number of drugs that were introduced into the market per $1 billion. And we can see a log linear relationship. Each year, there are fewer drugs introduced into the market per $1 billion. It was named Erum law, which is the inverse of Moore law, the law in computer science that states that computing becomes cheaper every year. It's the opposite in drug discovery. There are several reasons for that. One of that is that just FDA becomes more and more stringent in terms of what drugs are introduced into the market or allowed to be sold on the market. However, today I would like to tell you a bit about how organic chemistry and difficulty of making molecules in the laboratory is an important reason that drug discovery is costly and expensive. So for that, I wanted to share with you a story of how penicillin was discovered, synthesized, and how it led to novel drugs in the end. So in 1928, Alexander Fleming has discovered penicillin. He was studying mold, noticed that bacteria do not grow next to a certain bacteria. And the structure of penicillin is drawn on the right. It's a bunch of atoms connected together. It's around 20, 30 atoms. And we will be mostly interested in one part of this molecule called the beta-lactam ring, these 4 atoms also connected to an oxygen. But first, few words about how to read molecules. It's nothing to be scared about. I am not a chemist as well. So we will be using a simplified notation. So the beta-lactam ring is shown on the left in the simplified notation. But if you were to draw each individual atom, the beta-lactam ring that's part of penicillin has 3 carbon atoms. It has an oxygen atom. It has a nitrogen atom, and then it has 5 hydrogen atoms. But chemists are, lazy, and they are not drawing all of these carbons and most of the hydrogens, and we'll be using simplified notation in this talk. Can I point? Fabulous. Okay. So in 1943, due to prolonged Second World War, the US and its allies have decided to try to make penicillin at scale and cheaply. while Alexander Fleming has discovered penicillin, it was produced by mold, and at the time it was too expensive to support war efforts. It took 2 years to produce the first synthetic or artificial penicillin, meaning to synthesize penicillin in laboratory without mold. However, the route was highly impractical. So it was below 0.1% yield, meaning that only less than 0.1% of each gram of substrates were converted into penicillin. This was achieved by Merck, but it hasn't changed how penicillin was produced during the Second World War because it was still too expensive. It took next 12 years to make penicillin cheaply in the laboratory. This was achieved in 1957 by John Shinnan. He before that discovered a few things about organic chemistry, and then he was able to make penicillin with reasonable yield, around 10%, in the laboratory. By that time, it wasn't too impactful because we already knew how to make penicillin cheaply using mold. So it seemed like it wasn't really necessary, more of a scientific achievement. However, in 1980, the advances in organic chemistry and the fact they were able to make penicillin artificially in the lab led to the discovery of imipenem, which is an antibiotic that is sold by Merck to this date and is quite useful. But first, a few more words about how penicillin works actually. What is the mechanism of action, and how this mechanism of action is linked to the fact that it was difficult to synthesize. So that's the beta-lactam ring that's part of penicillin. I'm not showing the whole structure, just these few atoms that make the ring. So beta-lactam ring is unstable. The bond's length and bond angles are just unnatural. They don't want to be in these configurations. But that's actually good for how penicillin works. Because penicillin, thanks to the fact that this ring is very unstable, can bind to an enzyme that's building bacteria wall and hence render bacteria inactive. On the other hand, because it is so unstable, it's just hard to put together these atoms together in the laboratory and make penicillin. So it's like both good and bad. And that's one of the reasons it took so long to synthesize penicillin in the laboratory. So a bit more about the mechanism of action. So we have here this part of penicillin, the beta-lactam ring. And here's enzyme. Enzymes are proteins. We have a lot of enzymes in our bodies. This particular enzyme builds bacteria wall. So we don't want this enzyme to be able to do so. And the way penicillin works is that this beta-lactam ring grabs the enzyme. So perhaps you can see that it extends one, you could say, hand and grabs enzyme. And because the enzyme is bound to the penicillin, to the beta-lactam ring, it cannot build bacteria wall. You could think about this as a mousetrap. So a mousetrap has several key components. One of them is bait, and another one is spring that opens and captures mouse. And beta-lactam ring is the spring. You might be wondering, well, what is the bait then? It's not very important, but the bait is the other part of the molecule that attracts enzymes. So enzymes gets closer, and then snap, it gets captured by the beta-lactam ring. So in 1957, we were able to synthesize penicillin, but how does it connect to the imipenem and the discovery of this antibiotic? So somewhere between the 1957 and 1980, scientists have isolated this compound. It's another antibiotic, it's not penicillin. It was very potent, but had one issue, maybe more, but at least one, which was that it disintegrated in water and in blood, so you couldn't really administer it as an active antibiotic. But thanks to the fact that they were able to make penicillin at scale, they were also able to synthesize analogs or different compounds that are similar in structure, but in this case are more stable. So in 1980, thanks to advances in organic chemistry, they were able to make imipenem. So to zoom out, we went from 1928, when penicillin was discovered, through the first efforts to synthesize it, to discovery of imipenem. And throughout the story, organic chemistry and synthesis planning more broadly was quite important. And because of that, and this is an argument why it is important and in drug discovery process and how it slows down drug discovery process, because it took like 12 years to synthesize penicillin cheaply. You might wonder if that's an issue that, it was in the past. Perhaps now organic chemistry is no longer a difficult part of drug discovery process. Just the opposite. As our new drugs are, more and more involved, we are more and more ambitious about what we want to synthesize, and organic chemistry remains an important roadblock. So here's a quote from a recent paper by Merck. They say, a recent examination of our notebooks shows that CN coupling is a hit or miss affair. 10,000 examples with 35% failure rate. That despite this potential to render drug-like molecules may be underused because of its higher demand for optimization. What it says is that there is this important organic reaction called CN coupling, and chemists, well-intentioned, go into laboratory and every day fail to perform it in quite a substantial number of times or percentage of times. It's similar to, as if GCC or your favorite compiler would fail for 55% of otherwise correct source programs. That would be quite annoying, and this is the reality we are dealing with in drug discovery. So to summarize this first part, the key points that I wanted to share, I know there are probably quite a few new concepts, is that drug discovery is slow and quite expensive. It can take 12 years to introduce a drug to the market and cost over $2 billion. Another thing that's important to keep in mind for the rest of the talk is that advances in organic chemistry translate to novel drugs, and also the fact that organic chemistry is difficult slows down the whole process. So it's like both. And in particular, I have shared with you the story of how discovery of penicillin and its synthesis led to a practical drug such as imipenem. And finally, I also mentioned how organic chemistry remains to be an important bottleneck. So this is the first part. I don't know if anyone has questions. I'm happy to take if something was unclear. If not, I will go to the second part. The talk is a bit shorter, so we have time for questions. So the second part is about how artificial intelligence and more broadly computer science contributes to speeding up drug discovery, but by making it easier to synthesize molecules. I will focus on one particular application, which is automated synthesis planning. It's software solution that presents chemists with ideas for how to make molecules in the laboratory. But for that, I need some nomenclature. So synthesis plan is a recipe for how to make a molecule. It's quite similar to a cooking recipe. So we want to have a recipe that allows us to go from things we can buy in a grocery shop, like rice or beans, and then sequence of steps how to mix them together to get what we want, a cake or a dish. So similarly, let's say we are starting with this compound here. I will refer to this as the target compound. So again, it is in the simplified notation, and well, we will make this molecule in a sequence of steps. So the first step, each step will be a reaction, just like in cooking. You can mix something, and under proper conditions, hopefully translates into a dish you were hoping for. Similarly here, the synthesis plan says that if we mix the two compounds on the right, I will refer to them as substrates. Under proper conditions, we get the product on the left. And they are color-coded. So you can see that this is kind of like breaking a molecule. So you cut this molecule in half, and you have the first half here and the second half here. However, these compounds cannot be yet bought on the market. They are not like rice or bean. We have to, let's say, break them further. So, we break down recursively, so this should be a familiar concept, molecule into these compounds. So this compound is simplified or broke down into this compound. And there is a tiny green circle. This indicates we can buy this compound on the market, so we don't have to break it down further. Similarly here, but this one is not yet available. We cannot buy it from any provider. So you have to break it down further. And this is a full synthesis pathway. If you were to give it to a chemist, he could buy these, order them online. These compounds would arrive packaged into a laboratory, and he could mix them together, react them together, and under proper conditions, hopefully get this compound. One detail to note, a cooking recipe is good if it's cheap. And similarly here, this compound was already possible or available on the market. However, the synthesis plan breaks it down further because these two were very cheap. So it was better to break it down further. So we know what is a synthesis plan. Now I wanted to talk a bit about how computers can design synthesis pathways like this. I don't know, 20, 30 years ago, this was done only by chemists manually. Right now it's often done together with computers. So how can computers plan synthesis pathways? It's actually quite similar to how computers can play games. So imagine you are playing a game of tic-tac-toe. That's your move. You are playing the O's. And a quick examination of this board shows that the optimal move is to play in the middle because otherwise the X can win. So it's a pretty simple reasoning process for us, humans. How can we make computers to do the same? So one approach, and this algorithm is called minimax, is to compute the whole tree of possible virtual moves. So the leftmost branch corresponds to the optimal move, as we already know. Now the center branch and the right branch correspond to playing in the corners. And these, by examining the tree, we can see that these moves are suboptimal because if we play here, the X can play such that the root of the tree corresponds to a losing position. So by working backwards, you could say, like you could use dynamic programming here, let's say, we can work out that the optimal move corresponds to the leftmost branch. So that's tic-tac-toe, but how about synthesis planning? So synthesis planning can be seen, and it's actually, that's how it's solved by some companies, including one I am working at, can be seen as a game. So we have to make a connection between games and synthesis planning. So first, an unfinished board in tic-tac-toe or in chess or Go will correspond to a synthesis plan where not all compounds can be at both. It's not a full cooking recipe, if you will. Now, a finished board, so here we can clearly see who won. That's the O's have won because they have the diagonal. Corresponds to a synthesis plan where each compound can be bought on the market. We don't know yet if this is the cheapest route or the most correct, but at least it is finished. Like you can give it to a chemist and hopefully he could carry it out in a laboratory. Now last but definitely not least, this is actually the most interesting part. Each move in this game is breaking down the molecule, if you remember. So we cut usually molecule into simpler components. It's the most interesting part because in contrast to tic-tac-toe, Go, or chess, we don't know which moves are correct or legal. So in chess, we know how the bishop moves, how the pawn moves. In organic chemistry, we don't know which reactions will work in the laboratory. We have textbooks, we have experiments, but we cannot say for sure, usually. There are no rules that encode chemistry completely. Unfortunately, that would be terrific if there were rules like that. And if you're interested to learn more, there is a great paper called entitled Planning Chemical Synthesis with Deep Neural Networks and Symbolic AI in Nature that gives a much better description and much more thorough of how computers can solve automated synthesis, can do automated synthesis planning. Now, as I mentioned, we don't know which moves are legal. And to determine which moves we can take, and by move here, I mean an organic reaction, we use for that deep learning. And I will share more details in the third part, but for now, let me just stop at this. So this is a legal move, meaning that if you mix together the two compounds on the right, under proper conditions, hopefully we get the compound on the left, right? So this is a move that we can execute in this imaginary game. Now, in contrast, this move is illegal. I don't know if you can see the difference. We have added a few atoms, you could say, to both compounds. To the target compound and one of the substrates. And this makes this reaction incorrect. It wouldn't work in the laboratory, and hence we cannot use it in this imaginary game. So if we were to mix together these two compounds, we would actually get a different product. And as I was saying, one approach is to use neural networks that takes as input a molecule, and as output, they give molecules that when mixed together give the target molecule. So they are predicting moves in this game. It's similar as if you had a neural network that predicts how a bishop can move in chess. It's an obvious problem, but in organic chemistry, it's not obvious to say which reactions are correct and which are not. And that's why we are using neural networks. That's why we are using neural networks for this problem. So just as a summary, two components, search algorithm that uses deep learning for moves. And the search algorithm, like the one for tic-tac-toe, searches through all the possible reactions or ways to break molecule and looks for the way that results in the cheapest possible route. Well, it's natural to think, well, we have replaced chemists. It's not the case. And it's a useful story it's a useful concept to share because I think it is similar in other domains. So, AI won't replace radiologists reading breast cancer screens, mammograms, and in other domains as well. But I think that, and that's paraphrasing smarter people than myself, I think that AI won't replace chemists, but chemists who use AI will replace chemists who don't. So chemists and computers have complementary strengths. One breakdown is that chemists take a long time to design a single synthesis plan. It's very difficult to put a number to this, but it's probably over 1 minute at least. In contrast, computers don't get tired, so they can produce as many synthesis pathways as you want, assuming you can pay for compute. Chemists are much more accurate, and they are not only much more accurate, they can invent novel chemistry. Computers and artificial intelligence, not so much. The reactions will be much more often incorrect and much less creative in terms of the chemistry concepts used. And finally, chemists do not remember prices of millions or billions of compounds, while computers can store them in a database. And it is an important component because you're looking for a synthesis pathway that is cheap and that you can order on the market, where you can order all ingredients or substrates on the market. So you can combine the two. And chemists can use computers, for example, to look through different ideas and just pick the best one. And similarly, probably in breast cancer screening, maybe radiologists might use AI to highlight suspicious regions, but defer to their own judgment in the end. In the final part of this, in the final few slides of the second part, I wanted to describe one specific use case where automated synthesis planning is already speeding up drug discovery and making a difference. So this is in de novo drug discovery. De novo drug discovery is about, it's a type of drug discovery process where you are starting from molecules that are already novel. No one has seen them before, perhaps. And because you're starting from novel molecules, you have to tell which one are possible to synthesize because you cannot make any molecule in the laboratory. De novo drug discovery, I guess you could say it's becoming more and more popular due to advent of generative neural networks. Perhaps you have seen neural networks that generate images, that generate text. You can also make neural networks that generate molecules. For example, you could encode a molecule as a sequence of characters, and then a neural network outputs a sequence of characters that are then interpreted as molecules. And I also, at the bottom of the slide, linked a paper generating focused molecule libraries for de novo drug discovery, if you are interested to learn more. It's quite interesting how in deep learning you can repurpose often models from one domain to another. So why is synthesis planning a bottleneck in some applications in de novo drug discovery? There are just so many drugs, so many molecules that might be drugs. So it's hard to put a number to this again, but one estimate is that there are between 10 to the power of 20 to 10 to the power of 63 potential drug-like molecules that have been that are possible to synthesize potentially, while only 1 billion of them have been actually made in the laboratory. So there is this whole universe of possible molecules, and we just don't know a priori which ones of them we can actually make in the laboratory. So we probably have to use algorithms to decide, because no chemist can screen such an astronomical number of molecules. To illustrate this, one concrete use case, we were, as a company I am working in, we are collaborating with University of Montreal on a project related to pandemic. So they were looking for molecules that bind to protein in SARS-CoV-2, but they were looking through a very large space of molecules. Their search space consisted of 10 to the power 18 molecules or something like that. So it was infeasible to use even the fastest software for planning synthesis pathways. And in this space, there were molecules that differed quite little, at least for me. So here are two examples from this collaboration. These are two molecules, again, in the simplified notation. And they both have high affinity to the studied protein. It's measured in something called binding energy. It's not very important. But what is important is this one is hard to synthesize. This one is easy to synthesize. And the goal is to discriminate between the two, because, well, we have to make these molecules in the laboratory to study them. So as I was saying, even the fastest approaches in the class I was mentioning were too slow for this use case. So it takes on a single CPU to play this game to the end. Like, it's hard again to estimate this, but probably usually over 1 minute on a single CPU thread. So we had to remove search from the equation. And what we did is we pre-computed outputs of the software for a large number of molecules in the first stage. And then we trained a neural network to predict whether or not a molecule is easy to synthesize for a novel molecule without the search component. So we went from 200 seconds per molecule to below 1 millisecond per molecule, thanks to removing the search component. But it's difficult to do so without affecting accuracy, and it's definitely a work in progress. The way we validated these approaches is we took 3 search spaces, or more precisely, our collaborators took, and in each search space, we computed scores using our software, then, well, using the approximating neural network, and we saw a pretty good agreement in both search spaces. They differ in terms of complexity from the least complex to the most complex. However, anyway, in each search space, there were many compounds that were scored as easy to synthesize, but were hard to synthesize, and vice versa. And that's why it's still work in progress. So this concludes second part. And key, again, I know many new concepts, so key things to keep in mind for the final part. A synthesis plan is like a cooking recipe, a recipe for how to make a molecule in a laboratory. And to automate this process, we can combine artificial neural networks with search algorithms like ones used to play games. And deep learning is used to predict which moves are legal in this imaginary game, while search is used to look through all of the different possible moves, just like in a game of chess or Go. And this software already complements how chemists work. It won't replace anytime soon chemists, but it does already contribute and improve their work. And I have shared one specific example in something called de novo drug discovery, where we are potentially looking through a universe of possible molecules, and there are just so many that we have to use computers to look through these ones that we can make in a laboratory. So again, I don't know if anyone has a question, happy to take it. If not, we can proceed to the third part. Okay. I think we can then go to the final part. So I talked a bit about drug discovery and quite broad concepts in drug discovery, then a bit about automated synthesis planning. In the final part, I want to zoom into the deep learning part of the equation, and also highlight in the process of doing so, what are the current challenges? There are still so many things, to build and to do. And let me put here a small comment. I do focus on artificial intelligence, but there are many things to, again, build and solve that are, more broadly in computer science. So UX, databases. It's not only artificial intelligence. So what's the challenge? Synthesis planning can be seen like a more difficult version of Go. If you don't know the game of Go, you can think also about chess. So the goal is to find a cheap synthesis pathway, a cheap cooking recipe. And for that, we have pretty good algorithms to do so. It's, of course, a very difficult problem, but let's say we can use something off the shelf. I haven't talked in detail about this, but we can use things like A-star or MCTS, and these work quite well. The key challenge, at least in my opinion, is that we don't know how organic chemistry work, or we don't know which moves are legal and which are not. We have to approximate organic chemistry. And that's a challenge. There are, I think, two key reasons. First of all, we do not have a simulator. It's natural to think that, well, why not just simulate outcomes of organic chemistry? Well, at least until we have quantum computers, this is infeasible in a practical sense. We could do it for very small reactions. And also, it's quite expensive to validate a reaction in a laboratory, and also takes some time. So we cannot, try everything in the laboratory if we want to solve synthesis planning. Second, we do not have that much data. So deep learning, and I will talk a bit about this as well, works well when we have a lot of data. But right now, we do not have yet a lot of data. It's hard to say how many reactions there are recorded in the industry, but perhaps if you were to aggregate all of the reactions that someone has noted down in large pharma, this could be like maybe 20 million. We use, at the company I'm working in, we use open access data sets, and that's around 1 million reactions. You might be interested to learn that we use reactions from the patent office, more precisely the US patent office. And the way it works is that people are patenting molecules, and in a patent like this, they are also disclosing how to make it in a laboratory. So you can scrape this data and then aggregate it and form a data set to train neural networks. However, it's still pretty small. It's just 1 million reactions. And what is small, what is large is always relative. One reference point is that AlphaGo, an algorithm used to play Go at a superhuman level, required billions of moves to learn to play optimally Go, or very well. Again, it's hard to make such comparisons, but one thing for Senten, 1 million reactions is a pretty small number for how complex organic chemistry is. And the reason it is also challenging is that we are in the business of making novel molecules. We are not interested, as the whole field, in making the next aspirin or something like that. We want to find a novel molecule in this universe of possible molecules, of possible drug-like molecules. And because of that, we have to predict chemistry not for known compounds, but for compounds that no one has ever seen or synthesized. When we are going into laboratory, we are making something no one has ever seen. And because of that, we need artificial intelligence and deep learning that works well on novel stuff. And that's a challenge. But let's take a step back and talk a bit about what is deep learning. So a deep neural network is a machine learning or computational model that is very roughly inspired by how the brain works. So an input image, for example, an input image is processed by a sequence of layers, just like an image is processed by, at least initially, by a sequence of layers of neurons in our visual processing system. Then the final neurons are interpreted as outputs of the neural network. So here we can see that the neural network tells us that this is a dog with 62% probability. That's incorrect, it's a cat. So the way we train these neural networks is that we nudge them and we change these connections a bit so that the next time the neural network is a bit less confident it is a dog and a bit more confident it is a cat. And the way we use deep learning, as I mentioned, or how generally automated synthesis planning, sorry for that, uses deep learning is that for each reaction or for each compound, a neural network is used to predict which compounds can make it in reverse. So it predicts which moves we can take in this imaginary game. And why should we use deep learning at all? Here's, I would say, a tongue-in-cheek, but also quite serious slide from Ilya Sutskever, who is chief scientist of OpenAI. We'll actually talk a bit about OpenAI towards the end. So what he said is that if you have a good and labeled data, and you train a very large neural network on this data, success is guaranteed, as he said. Of course, it's not always the case, but it's a nice framework to think about why and where you should use deep learning. So deep learning had its watershed moment in 2012. An ImageNet is a benchmark in computer vision. It's composed of around 10 million images of cats, dogs, and whatnot. And the goal is to classify what's the object in the image. And until 2011, approaches not based on neural networks were winning this competition, achieving like 26% error rate, the lower the better. Then in 2012, to a surprise of everyone, a neural network won the contest with 10% error rate reduction. And in 2015, a neural network beat an estimate of human error of 5%. And it did check many boxes. The application in computer vision, it was a very large data set, at least for the times. It consisted of 10 million examples. And indeed, when they trained a quite large neural network on this data set, it performs surprisingly well. But it's not all roses, and applying deep neural networks is very difficult in business when you are applying neural networks on novel inputs. And as I mentioned, in drug discovery, we want to synthesize novel molecules that no one has ever seen. So by definition, we are usually applying neural networks on novel inputs. So here's one example. It's not only that a neural network makes here a counterintuitive prediction, but one that is quite dangerous. So this is a computer from a Tesla car, and what happened is it mistook yellow moon for a yellow traffic light. So it was slamming on brakes on a highway because it was thinking that it's a yellow traffic light. It has probably never seen yellow moon in this context and was quite confused by it. There are many examples like this in deep learning, at least right now. As a field, we are making progress, but it's still a huge challenge. So here's another one. So we have a neural network trained on the aforementioned ImageNet benchmark, and we just ask what are the top 3 predictions for the image shown on the slide. So in this case, it's a close-up of elephant skin. And the neural network, I would say rightfully so, classifies it as Indian elephant. It assigns 82% confidence to this class. The next 2 predictions are Indri and black swan. So looks reasonable. Let's try something else. So this is an image of a cat, naturally, and the neural network predicts with 71% that this is a cat. It also assigns a quite large probability to a gray fox and another breed of cat, 3.3% to Siamese cat. But the neural network makes a quite counterintuitive prediction, at least for me. I will let you judge, when we overlay the 2 images. And it's also a novel input. So this image was not seen in the training set. I don't think it was in ImageNet. And similarly, we have issues with molecules that we have never seen in the training set. So in this case, you could say it is a conflict between a texture and shape, because the image consists of a cat. I mean, you can see the envelope of a cat, but you can also see texture of an elephant. So in this case, it assigns very high probability still to Indian elephant, to Indri, to black swan, so the same predictions as on the leftmost image, but the cat is nowhere to be seen for the neural network. And again, you wouldn't, I mean, at least I wouldn't be able to predict this output beforehand, and this makes applying deep neural networks in business quite challenging if you are applying it on novel inputs. So let's come back to chemistry. So perhaps you remember this reaction from the first part. So the thing is that if you mix together these two compounds on the right, you get the compound on the left. In this sense, it is a correct reaction. It's actually a quite simple reaction that's taught at the university level. Now, this is an incorrect reaction, meaning that if you were to mix the two compounds on the right, you wouldn't get the compound on the left. And we have added 3 atoms, or changed 3 atoms, right? We have added 1 carbon atom, 1 chlorine atom, and 1 oxygen atom. The 3 make a group that's called acyl chloride. And it's kind of similar to beta-lactam, if you remember, group. So it is quite unstable and reactive, which often changes outcomes of reactions. Now it's similar also to the situation with yellow moon or the cat and elephant skin, because such compounds, because they are unstable, are very rarely present in the training set. So we just ask the neural network to make prediction for an input that was quite novel in comparison with its training set. So to quantify this, what we did, we took a few groups like this. Acyl chloride is just one example. For each group, we made artificial reactions where we added this group to product and one of the substrates, just like in the slide before. And then we measured AUC, you can think about this as accuracy, for each of the groups. So we tested 10 groups, and the higher the better. And you can see that in all cases, it's below 100%. So we were able to fool the neural network in all cases, and in particular for one group, not acyl chloride, but quite similarly acyl fluoride, the neural network in 40% of cases, roughly, it's AUC, not accuracy, but roughly in 40% of cases, said that the reaction is okay, while actually it was quite incorrect, and you could even say offensive for a chemist. So if chemists were to look at a reaction like this, he would be, you could say, terrified, because that's a quite unstable or reactive functional group, and generally you wouldn't want to perform a reaction like this, at least not, without care. So it's to close with a hint of a solution. So we do not know yet exactly how to solve this in chemistry, generally. There's a lot of progress, of course. However, there has been tremendous progress in computer vision, maybe to a lesser extent, but also in natural language processing. So ImageNet consisted of 14 million examples. That seems large. But if we follow the idea to train neural networks on larger and larger data sets, because this makes success more and more guaranteed, if you will, here researchers from OpenAI, the company I mentioned, aggregated a data set of 400 million examples based on images from the web. So what they did is actually a plot from, I believe, Google, because they did concurrently a similar idea. They executed a similar idea. So what you can do is you can collect automatically from the web images and the associated labels or titles of images. You can, for example, use alt text on web, right? So just that small text that shows up when you hover mouse over an image. You can use this to train neural networks, and you can see that they can be quite noisy, but also very useful for training neural networks. And web is infinite approximately, so you can gather a lot of images like this. So then you train a neural network to assign which label came from which image. So in this case, there is an image of a photo of a bed next to a bird feeder, and the neural network is tasked on assigning high probability that this image comes from this label as opposed to other labels that are less descriptive in this case. So what you can do is, I mean, what researchers did in the paper, they compared accuracy of the neural network on many data sets, but here I will be showing results on 3 of them. So again, ImageNet. ImageNet consists of many images of dogs, cats, but there are also bananas. So these are, quite typical images of bananas shown as examples. ImageNet has more examples than that. And the model trained on ImageNet got 76% accuracy, while model trained on the web scale data, also with the new methodology, got similar accuracy. It's probably by accident, also 76.2%, so nothing interesting yet. However, now they evaluated models on different data sets that were different from ImageNet, just like Yellow Moon. They were inputs that were novel to the neural network. For example, there is a data set called ImageNet Sketch that consists of many objects, but they look like as if they were hand-drawn. I'm not sure if they were, maybe they were. So the model on ImageNet gets 25%, while Clip, which is the OpenAI model, got 60.2%. It's a huge difference of 35% accuracy points. And the final data set was the hardest that I wanted to show. It's called ImageNet Adversarial, and it consists of images of objects that are very confusing in some way. So, for example, you might have a banana that's held at a very weird angle, such that you cannot see the curvature. Or you can have banana next to other objects. So here is a small banana next to, I think, sheets of paper. So the ImageNet model gets 2.7% accuracy. It makes mistakes very often, let's say, while the the Clip model gets 77.1%. So I think the lesson here is that one way to make neural networks more reliable is to just get more data. It sounds simple, but it's a very powerful concept, I believe. You cannot always get more data, which can be an issue. So that's everything I wanted to say. A quick summary of the main things. So in the first part, I mentioned that, or tried to convince you that synthesis planning is a key bottleneck in drug discovery. And on the flip side, advances in organic chemistry lead to novel drugs, such as imipenem and many, many more. In the second part, I described one specific solution to address this bottleneck. More precisely, I talked a bit about automated synthesis planning. So synthesis plan is like a cooking recipe, and computers can be used to plan how to synthesize molecules in the laboratory. One approach is using AI and deep learning, where AI is used to play the game, right? And deep learning is used to decide rules of the game. And I talked a bit as well about de novo drug discovery, where we are looking through potentially very large numbers of molecules, and we have to decide which one are possible to make in the laboratory. And finally, I also wanted to share a bit about how the field is evolving and what are challenges that we face every day. In particular, deep neural networks cannot be really trusted, let's say, and we have to put a lot of care into using them correctly. And I shared a few examples, such as the Yellow Moon example, or how the neural network reacts to weird images. But finally and most importantly, I hope that this will inspire you to learn more about how computer science can be used in drug discovery, and perhaps even get involved, because we need in this field more smart and motivated people like yourself. So that's all. Thank you for your attention.
Any questions? Okay.
Hello, hello. Oh, działa.
Hello, so I have a question about, you said that AI is used to to discover new chemistry, like I forget the word, you know, the connections, yeah? So, and you said there are certain rules that are then checked in laboratory. So are there any rules that are illegal for AI, but those reactions may occur in real chemistry in laboratory?
Let me try to rephrase to see if I understand correctly. So you're asking if we exclude some reactions from our computer, like from our software, or generally software in the space, because we don't want people to use them in the laboratory.
No, because we think they are illegal, they won't work, and we exclude them.
So generally the goal of these, of the deep learning part, the two parts, one of them was deep learning, was to learn which one do work in the laboratory. So that's the learning goal of the algorithm, of the neural network. But quite often they make, let's say, incorrect predictions, and you have to, there's one approach, you don't have to, but one approach is to correct them a bit manually. So you can encode, for example, manually, that you should never predict that a reaction would happen if there is, this acyl chloride, if there are some atoms that are very highly reactive, because it could be dangerous for people, or maybe you do not want to make predictions and suggest reactions that have to be performed in very stringent conditions. So, for example, they produce fumes that are toxic for humans. So yes, you do sometimes things like that. There are different approaches. You can either do it more manually, so just have if statements, if you will, in software to say, okay, if there is this group, just don't show this reaction. But you can also go more, let's say, be more end-to-end about this, and just add to your training set such examples and penalize neural network if it makes such predictions. It's similar to self-driving car industry, where there are different approaches. So some companies enforce safety of their software by encoding rules and safety breaks, and some are trying to be more end-to-end about this and try to have a single, let's say, neural network that just predicts the steering wheel move, and try to add to training set as many examples of incorrect behavior as possible.
Okay, thank you.
Thanks for the question.
Okay, so this is the last question.
So you said there are so many reactions of these molecules. So can you tell me, it is possible that neural network accidentally creates some very dangerous reaction or poisoning, and chemists won't notice? Are you prepared for this in laboratory?
Right. That's a very interesting question, especially with applications of this technology to more and more automated processes, where we can rely less and less on chemists. But right now, usually the way these softwares like this are used in drug discovery is more manual. So in the end, it's the chemist making predictions. Again, to use the analogy of self-driving cars, we do not have yet full self-driving car autonomy, and similarly, I don't think there is a single laboratory right now that's fully driven by predictions made by models. But thank you, very, very good question.
Okay, so massive thanks for Stanisław for showing us how computer science helpful can be. So the next lecture is about big data, Apache Spark. Stay with us.